Hello and welcome. I am Miss Stephanie, and in this video we are going to learn about Punnett squares. Punnett squares are used in genetics to determine the possible combinations of genes that will occur at fertilization. It consists of squares, which are used to create a combination of genes and predict the possible genotypes of the offspring. In this video, we will only learn about monohybrid crosses, and in a subsequent video, we will talk about dihybrid crosses. A monohybrid cross is a cross between a single paternal and a single maternal gene. For our example, we are going to use height as our gene. Tall will be the dominant trait, represented by a big T, and short will be the recessive trait, represented by a little t. Before we can start talking about how to do Punnett squares, we need to understand a few terms first. The first term we need to know is genotype. A genotype is the letter representation of a person's gene. The genotype is consisted of two letters, either lowercase, uppercase, or a combo of the two. The next term is phenotype. The phenotype is the physical representation of a genotype, so being tall, or the color of your hair. The next word is dominant trait. A dominant trait is one that only needs one allele present for its corresponding phenotype to be observed. A dominant trait is represented by a capital letter in the genotype. With our example of height, tall is our dominant trait represented by a capital T. The letter used to represent a gene is typically the first letter of the dominant trait. The next term is a recessive trait. A recessive trait is one that needs both alleles present in the gene to be recessive for its corresponding phenotype to be observed. A recessive trait is represented by a lowercase letter
powered by video.